All right, let's go over chapter one. Um, the chapter one in A&P courses is really to set the stage of the course and just give you sort of an overview of some different topics that are kind of important. The other thing that often comes up in chapter one is directional terms and anatomical terms. And that's actually dealt with separately in this course. So it's called Atlas A. So if you don't have the book that, and by the way, reminder, Dr. Tice likes that one book to be used for AMP2. So when it talks about Atlas A, it's referring to that particular book that she has. So if you're taking AMP2, you'll have to get that eventually. In my case, there's a separate PowerPoint and video for the major directional terms and anatomical terms. And the ones that you should know, you'll see in the study guide for the first exam. Those, like I said, in the video that you have to watch for the other one, no, you don't have to watch, but it's up to you whether you watch it or not. But these terms are just, they're literally things you need to memorize. I'm not a big fan of flashcards, but in that case, since you just literally need to memorize these terms to function well in anatomy and physiology, that might work. So you need to watch that video, it's separate. And then the rest of the material that is traditionally in chapter one is found here. And I'm gonna go over that now. All right. Now, so here we have these major themes, all right? So that's what this is about, not directional terms or anatomical terms. Again, that's a separate video. So themes, and in the picture I chose here, like, I don't know if you know this, but initially, people were not allowed to study anatomy. And what physicians would do is that they would, this sounds really morbid, but they would find cadavers or dig up people who were recently deceased and then dissect them so that they could learn about the body. And you know, I'm not saying that's what's happening in this painting, but this is a really early study of anatomy. You can see they're not using a human in this case, but very often this was done at night. Apparently, that's what I was taught, anyways. As opposed to now, when people donate their bodies to science and other things, and going to see body works or whatever, cadavers dissected in interesting ways. But that's just to sort of get us going. Now, again, and you'll see with every chapter, you've got your learning outcomes for the course. These are the same if you're here or in Dr. Tice's class, again. So physiology, and you're gonna find it's to really study physiology, you really need to know a lot. And that's why the next chapter, which heads up is huge, is looking at chemistry. And I'll talk about that in that chapter. But the reason I mean, it's always in an A&P course, and especially here because a lot of students don't take chemistry. So we need to fill you in, and that is why chapter 2 is probably the scariest chapter in this whole course. The other one that will probably scare you is an AMP2, the endocrine system. I don't know why, but students have trouble with that chapter. So we're going to go over chemistry. And the reason that is that's sort of the bedrock of the other life sciences, right? So you have chemistry and molecular biology, and that combines with the studies of atoms, molecules, cells. Then you're going to get into tissues, and then obviously we're going to do all the organ systems throughout the year. When you go on to AMP2, you'll do the rest of the organ systems. And these ideas here, populations, ecosystems, and biosphere, you have to take ecology classes to study that part. We sort of get to here. And then when we're talking about organism, we're focused on human anatomy, but there are anatomy courses that don't. So for example, the equine students take equine anatomy. And I used to teach my 105 class right before the equine students came in for their anatomy class. So it was really quite interesting because they were often very in the same place in their course as I was, only I was doing human and they were doing horses. But anyway, so obviously we're human-centric. And it's really important to understand that this is why you need to have some chemistry. Otherwise, you're not going to really appreciate how these things function. And if you can't appreciate how these things function, you don't understand how drugs function. And if you don't understand how drugs function, if you're going to the clinic, you're kind of useless, to be honest. And that's why mistakes happen, right? So, I mean, it's up to you. The more you learn and the more you understand, the better practitioner you're going to be, all right? And that's really how it all works, right? Same thing with professors. The more they know, the better professor they are. If they don't want to learn anymore, they're not as good. So anyhow, there you go. So this is what we're going to be doing. And in terms of studying anatomy and physiology, you're supposed to understand the difference between physiology and anatomy. So anatomy is the form. So how do we study form? Well, dissection, comparative anatomy, which I love. When I was in school, my anatomy course was an animal anatomy course because I was focused on learning laboratory animals, and this was a lot of fun. Exploratory surgeries, medical imaging, we'll talk a bit about that, and gross anatomy. And then at the microscopic level, histology, that's looking at tissues. So histopathology, so what's going wrong with tissues, cytology, cells, ultrastructures. But this is all form. Now, in physiology, anatomy and physiology, the reason you take them both is because form and function are related, okay? So physiology then is a study of function. So there's a lot of subdisciplines in this because it's really quite complicated, so people get very specialized in their particular ologies. So there's neurophysiology, is the study of the neurons and how they function. Endocrinology, for example, hormones. Pathophysiology, related to disease. And then you can study different types of species and how things function the same or different between them. And that's really useful for understanding human physiology, right? So for me, my laboratory specimens are planarians, which are flatworms, but a lot of the genes they have are the same as the ones we have. And we can test drugs on them and look how cancer develops in them, and get some understanding when we go back and look at human, all right? So so again, anatomy is structure, physiology is function and how that works, right? And they're related because the form and the function are, obviously intertwined, okay? Now, let me just look, make sure. I think I had another thought in my, oh no, it's the next part coming up. All right, so then there's the hierarchy of complexity. So it depends, just make sure you keep these ideas in mind. I usually like going from small to big, like over here. And this slide I actually grabbed from Dr. Tice, which is the opposite to what I usually do, and maybe she likes thinking about it the other way. So you should be able to do it both ways, right? So the way Dr. Borer likes doing from small to big, or the way Dr. Tice likes to do it from big to small. But understand, I'm going to start at number 7 just because for some reason my brain works that way. The smallest part that we're concerned with in A&P is the atom and how they go into molecules and form bonds, and then how all of those molecules form the organelles that you find in your cells, and then the cells that are required to make up a tissue. And the tissues then are combined to make organs. And then the organs are part of systems, so organ systems, and that all together makes up the organism, all right? Now, it's really important to understand when you're looking at human anatomy or any kind of animal anatomy for that regard, is that even though we are sort of the same, there are variations. And that is why medicine is an interesting field. And it's also why it makes it difficult to just say one size fits all, and why when you do clinical trials, some people are more receptive to certain treatments than others, all right? Because while we all might have kidney and heart, they don't look always the same, okay? So when you're learning about the form, that would be what you see most of the time, but there's variations on that. So you can sort of see this. You're going to learn what all these vessels are next semester. Look at the kidneys here. So here's the adrenal gland, kidney. Actually, I only have one kidney. This is actually what I got, cross-fused kidney. I have one large kidney in the lower back. It looks pretty much like this, but apparently my tubing is all kind of weird looking, all right? So over here is what you classically see, all right, which is what most people have, but there can be variations, all right? So this is very important to know, all right? And for me, during this COVID time, apparently people who had COVID have a lot of kidney problems, and some of them end up on dialysis for life. So I've been told I have to be very careful because I've only got a kidney like this. So if I damage it, it's not like I have another one that I can rely on. All right. So, things to keep in mind. All right. No two humans are identical. Now, when it comes to the characteristics of life, you've probably seen that before, but it relates here. There's 8. So we're going to talk a lot about all of these, right? So there's the 4 biological molecules that are important for living things. So that would be your carbohydrates, your lipids, your proteins, and your nucleic acids, all right? All living things require energy and materials to maintain life, all right? And then in my notes, I talk about, well, we get our energy from food. The energy is released from the bonds that are broken down from that food, and then the molecules and the atoms are then rearranged and stored in different ways. We have ATP, or energy currency, or energy transfer molecule. It's not an energy storage molecule. That's your fats and lipids, but it's the way we transfer energy from one place to another. Metabolism. Oh, wait, I'm skipping here. Composed of cells, all right? So the cell is the smallest unit of life because it has all 8 characteristics of life, all right? So viruses, not technically living, all right? I know that sounds pretty wild, but they're not, because they don't have all of these 8 characteristics, all right? They're smaller than cells. They don't have the ability to do this on their own, which is number 7, all right? So we'll get to that in a sec, all right? So just wanted you to know. So if people say they're going to kill coronavirus, can't kill something that isn't living. So the proper way really is to say you're going to inactivate it, all right? Okay, now metabolism and excretion. So metabolism, as we'll go over, is composed of anabolism, which is building, and catabolism, which is breaking down. And there's wastes that are associated with this process. Water, CO2 are typical wastes that we think of, and other things. So you'll end up talking about that. Responsiveness and movement, all right, so responding to the environment. When I teach 105, I show a picture of a gecko. Oh, no, sorry, chameleon, not a gecko. A chameleon, right? So remember, they can change their color to respond to the environment. Movement, can move around. Or have things that move. All right, I know that sounds weird, but not all cells move, for example, but they move in their own way. So this doesn't necessarily mean walking across a room. Homeostasis, all right, so this is the desire to keep a certain balance, all right, we'll talk about that in a sec. Reproduce and develop, and again, remember that doesn't apply to viruses. They can't do that on their own. They require other cells to do that for them, and that's why they are so dangerous. And with reproduction and development, you have to think about differentiation, right? So if you think of a butterfly, it changes its form from the insect to in the form of the insect, right? So sometimes it's caterpillar, then it's butterfly. Growth, right? So you know that you're bigger than you are now than when you were a baby, so you grew. The other thing is really critical is evolve. Viruses not living things, but boy, can they ever evolve. They do that real quick. But evolution, as a human species, we evolve to suit what's happening. Eventually, for example, there will be people that are immune to coronavirus, right? So we're not right now, but eventually. But evolution on our scale takes a long time, so it's hard for us to see it, but it happens. So yeah, and I put this picture here, like rock versus fish. I think a fish is alive, a rock is not. It's sort of oversimplification, but this fish has all of these 8 characteristics, all right? Okay, let's see if there's any thoughts that I put down in here. Yeah, number 8, it's important to remember the process that drives the evolution is called natural selection, all right? If you didn't learn that during your science background, you probably look it up, understand it. Yeah, all right. Now, there's 4 general themes in anatomy and physiology. I kind of want to go over those. So the first one I already started with, and that would be structure versus function. And usually, because we're human and we don't see really tiny things, it's usually what we think of, right? So the structure of the liver is associated to how the liver functions, all right? Liver is really fascinating. I don't know if you realize this, but the blood actually is circulating, it just literally is like bathing the liver, right? So the blood vessels there are really leaky. So that's really important because this is where you're making a lot of stuff and you're trying to get it out into the body. You're also making you're filtering here, and you're going to make your your bile here. What am I thinking? It's just not coming to my head. It's late today. And that's also being made here to help you digest your fats and get stored up in your gallbladder. But there's also the microscopic part with structure and function. And you'll see that when we do our chemistry chapter 2. For example, proteins have a very, very specific structure for their function. And even small little changes will affect that, right? So in my notes, I talk about hemoglobin and sickle cell anemia. With that, there's just one molecular change. When proteins are made up of amino acids, they just change one amino acid in that whole structure, and it completely changes the shape of the hemoglobin, so the structure, and then it doesn't function properly anymore, all right? So it doesn't necessarily have to be at the cell and tissue level. It can also be at the molecular level, right? We have other lists here, like Tay-Sachs disease and cancer and cystic fibrosis. There's molecules involved with that, and they get something that's different about them, and they don't function. All right. Yeah, here I have bile right here. Oh, my goodness. Anyhow, yes. And the other thing is energy, and we talk a lot about that, especially in chapter 2 and where we get it. But remember, the important thing here is that we're getting our energy from our food, and there's different ways that we extract that energy. And here you've got glycolysis, Krebs cycle, and oxidative phosphorylation. I'm actually not going to go over that with you, but you are expected to know it. You're not expected to know all the different steps and the different molecules involved, but you are expected to know different aspects of it, such as glycolysis takes place in the cytoplasm. It doesn't require oxygen, while as Krebs cycle and oxidative phosphorylation do require oxygen to take place in the mitochondria. And most of our ATP is produced by oxidative phosphorylation. The rest of them, a little bit of ATP, but you're making mostly NAD or NADH and FADH2 to carry your electrons over to here. So you do oxidative phosphorylation. That's where you make your ATP, most of it. All right. So if you can't remember things like that, I have a video in the next chapter that you should watch. It will help you remember. Okay. Now, the other thing that I'm highlighting here, and it's something we'll talk about later in the semester, is that, ATP is our energy currency, right? So our energy transfer molecule. And the body has a very different ways of using it and maintaining it, right? So you probably, we'll go over it again in chapter 2, but it's something that is recyclable, right? The energy is an ATP, and then you release that, you get ADP, but don't worry, we'll go over that. But what I want you to see here is this is something that happens in muscle. Since obviously there's a lot of activity in muscle, it needs lots of energy. It has a number of ways to try and keep maintaining a supply of ATP. It has its own ATP, but it also will allow for a recycling of the molecule using creatinine, or creatine, which we'll talk about later. And then, of course, it can generate, muscles can generate ATP in the absence of oxygen, and then in the presence of oxygen. In absence or presence. So absence where it says no, and where there is oxygen here. So here you can see your mitochondria, And here, this process is called fermentation, right? So you build up lactic acid and that sort of thing. But basically, the body is very creative in its ways to sustain its energy supply, all right? So we'll talk more about how that works later when we do muscle, all right? And then there's the flow of information. So there's this thing called the central dogma. I don't know if you've learned that. And if you can't remember how this works, this is another thing that I don't really cover in ANP1, all right? Again, it's one of those things that are thought that you should know. Sometimes it shows up in chapter 3, but the book that you guys are supposed to be using, it doesn't seem to be in there. So again, if you need a refresher on the central dogma, now what is that? Central dogma in terms of flow of information, right? So this is genetic information, DNA. Your DNA codes for your proteins, but the DNA is in the nucleus. So the RNA is the transfer molecule. It takes the messages or the coding for protein from the nucleus into the cytoplasm, where you're going to make your protein. So this is central dogma: DNA, RNA to protein. But dogma is something that is just like law, right? But as we learn more about science, we understood that there's this thing called the extended dogma. And you can go the other way. You can go from RNA to DNA, all right? So notice these are viruses. Very good at doing that. You can make DNA to DNA. And then in some cases, in the lab, you can go DNA to protein. So that's the extended dogma, right? So this would be special case, it's in red. Then there's chemical information, right? So this would be like hormones, signaling molecules, leukotrienes, things like that, which we'll talk about later. Really important chemical messages that are passed around the body. The other one would be neurotransmitters, right? Do I have a list of them in here? I'm just looking at my notes here. It's been a while. No, nope. I just had hormones and neurotransmitters, but there's more. Okay, so I'm looking at page 3. And then, of course, electrical information. All right, so that's the movement of charge. It goes over muscle and neuronal cells. So that's electrical information. And that information that can be passed along can be local or long distance. And that really refers back to this chemical part here, all right? Could be cell-cell communication or brain to the rest of the body. So it depends on how it works, right? So that could be electrical, right? So these things here is what we're talking about here. So localized or long distance, all right? Now, ultimately, what the body's trying to do is what's in theme 4, and that's maintain homeostasis, all right? So I've heard students tell me that they've had teachers that told them that homeostasis means equilibrium. No, okay? Don't say that, not equilibrium, all right? Equilibrium bad, equilibrium equal death, okay? What it means is balance, okay? Sort of a steady environment, a stable internal environment. It does not mean equilibrium, all right? And keep in mind that the human body is an open system. So there has to be a lot of ways of maintaining that. And that's why I like using this image here, right? So here you have your internal environment that needs to be in balance or relatively stable, all right? And that normal for one person, like again, remember we said every human is different. It's a little bit different than somebody else's normal, all right? And this is why it's good to go for a checkup every year, because they check and learn about what is your normal, so that when your normal is not normal, they know there's something wrong with you, all right? Okay, and when we say it's an open system, right? So food, salts, water go through the body, right? The digestive system. You inhale oxygen, you inhale air, you need the oxygen, exhale CO2. You got your circulatory system that takes stuff around, and then you're going to make waste, and you get them out of the body. So you can see that it's open, all right? It's not like you're a bag and nothing that goes is there that ever comes out, or nothing goes in, right? So the body has different ways of maintaining that balance. Now, what do I mean by it's not equilibrium? And this is what I mean, right? So take, for example, the ions in your body, all right? And they're separated by compartments. So here we're looking at compartment, the separating thing here would be the cell membrane, right? So what the sodium, chloride, and potassium concentrations on the outside of the cell, the extracellular fluid, very different than what it is on the inside, all right? And you use a lot of energy to maintain these differences, and these differences are called gradients, and they're used to do things in the body, all right? So they need to be maintained. If you're sick, you're going to have trouble maintaining these, and and eventually, upon death, they will equilibrate. So this is what I mean: equilibrium bad, balance good, equilibrium bad. All right. So try to remember that difference. Okay. In my notes, we say it's a dynamic steady state, right? Because things are moving. All right. So, and you have to remember these molecules and atoms and all of that are moving around between these compartments. So, so it's not like they're not in movements. We say it's a dynamic steady state. Okay. So the gradients have to be maintained. And how do we do that? Well, if you're an organism, you're in homeostasis. There's some sort of changes internally or externally. This causes some sort of internal change or a loss in homeostasis, and then you work to compensate for it. And if you can't, you get sick. And if you can, you're fine, all right? So this is what we talk about when we're trying to maintain homeostasis. Now, how does that work, right? So here we have this idea of mass balance. So in order for you to be essentially the same, the stuff that's going in has to be balanced by what's going out, all right? So this would be, a cistern that is, water's flowing in from, say, a river or somewhere, and then you've got to let it go out at the same rate. Otherwise, you'll overflow, for example. So you have to maintain this sort of balance in between what's going in and out. So the same thing with the body, right? So you've got your mass balance. So you've got your existing body load plus the stuff you're taking in and what you're pumping out, right? So excretion or metabolic removal, intake or metabolic production. And so for, say, water, like if you're using that, so you intake that through your intestine, lungs, skin, metabolic water. Again, if you look back at oxidative phosphorylation, you're making water there. And then you're going to output it through your kidneys, liver, and skin. Metabolism, you make that water into something new, all right? So very important to understand this idea, okay? So how does the body, which is an open system, do that, all right? So this is where we have these control systems, and there's a lot of them in the body, all right? So there's some sort of a signal that's put in as an input, and then an integrating center. And you might think, well, the integrating center is the brain. It's not always the brain, all right? So you really have to look at the system that you're considering to understand where is the information being processed. And then that integrating center is going to send out a signal, and then there's going to be a response, all right? So these control systems are really key to maintaining homeostasis in physiology, okay? So when you look at this, all right, and this is looking at something like temperature, say, for example, but say you have something that's going in, so you have some sort of an imbalance and a receptor is going to detect that, and that sends the information to the control center. And this is called the afferent pathway. It's always afferent. And then the control center is going to think about it and then pump out a response to an effector. And that is called the efferent pathway. Okay, so afferent has to come before efferent, A comes before E, okay? And then you're going to have some sort of effect that's going to bring you back to homeostasis, all right? Okay, so this is how most things are run in the body. And that's essentially what we have when we have this feedback mechanism, which is called negative feedback, all right? So negative feedback tries to compensate for a change by bringing it back to what it was before, all right? So it turns down or dials back some sort of an effect, all right? And the best way to think about this is like a thermostat on a fish tank, right? So you set your thermostat at 30, say, or if you set your thermostat in your body is 37, right? And then you'll sometimes be below that temperature, and then your body will do what it needs to do to bring it up, but it's not going to stop right at 30. You might go a little bit higher, and then it's going to go back lower, and then it has to do something that goes back higher. Maybe it overcompensates, and so on. All right. But basically, at the long and short of it, the average, or the, about where you are is, about 30. All right, or for the body, 37. All right. So this part here is what we call your normal range, right? And this could be a little different for different people, or different fish tank thermometers, right? But that will depend, okay? But this is, say, your homeostatic or your sweet spot where your body would like to be, or where the fish tank thermometer is going to keep your fish, okay? So this is negative feedback. So it's correcting for a change, trying to dial back or bring down. So that's why it's called negative. All right. The one that students think of right away when they think of negative feedback is temperature, right? Because you've got a different type of response, whether you're hot or you're cold, right? So you've got receptors in your skin that detect temperature. And if you're warm, right, since this case, the afferent pathway goes to the brain, your brain sends information back. And on the efferent pathway, you're going to sweat, right? And then it could be the opposite. Your body temperature could go down. So you've got cold receptors in your skin. The afferent pathway goes to the brain again. And the body is going to make you shiver and start warming up again. All right. But both cases are negative feedback. All right. Positive feedback. This is not as common in the body. There's some very specific examples of this. What's happening with positive feedback is that the effect is going to be augmented with time until the source of the effect is removed, right? So there's going to be constant increase or augmentation of the effect. So the only way to make it stop is to remove the thing that caused the change in the first place, all right? So there's a few classic examples. So blood clotting is one that affects both men and women, right? So basically there is a damage to a blood vessel, like here, and then hemostasis is going to be activated. We'll talk about the process involved in hemostasis later in the semester, but basically that's going to keep going and forming a clot here until the space is sealed, and then the system will stop. Okay, so that's positive feedback. The other two positive feedbacks that occur are female focus, so childbirth. So the baby pushes down on the cervix, and that causes the uterus to contract. Baby pushes down again, uterus contracts, and it gets more and more spectacular as the birth takes place, and it will only stop once the baby's born, right? The other one is milk letdown, right? The milk will come out until the baby stops suckling, all right? So that's also positive feedback. Then there's this other thing. Now, there's this, and I always use a picture of food here because it's really related to that. The next thing is not a feedback mechanism. It's called something. It's like it's called feed forward regulation, not feedback. All right. So don't say feed forward feedback. That's not what it is. I list it here because it's something that's also important to the body, and it's usually a good way to think about it is with food. Right. So this is all about anticipation and training. Okay. So classic examples is that if you even look at pictures of food or smell food, you might start to salivate, right? Because your body's getting ready to eat it. And similarly, your stomach will start producing acid, the sight or smell of food. It's not because you haven't even eaten anything yet. Your body just knows that you're going to get some, and it gets ready for that, right? The other thing is anticipation of exercise. If people are well trained, they'll begin to ventilate and lower their blood pressure in anticipation of a drop in oxygen and an increase in pressure and blood pressure for during the actual exercise. The other one, again, women focused, milk let down upon just hearing a baby's cry. Apparently that can even happen if it's not your baby. If you're lactating and you hear another baby cry, you'll let down milk. It can be quite embarrassing, apparently. I have a child that I wasn't able to lactate, so I don't know. This is what I'm told. So that's feed forward regulation, all right? Now, another one that you're gonna talk about when you do endocrine is insulin and glucagon. And this is why we just want to bring it up because it's kind of important. And basically, there's a feedback mechanism very similar to what we just did with temperature. And if you look at my notes on page 7, you can see how your body responds to blood sugar. All right. So, for example, if you have high blood sugar, what's going to happen is that insulin is going to dominate, and what you're going to do is take up that sugar, and you're going to work with the sugar to start storing it. You're going to convert it to our energy storage molecules, so glycogen and fat, and you're going to make proteins. And then when the sugar levels drop, you're going to use a different hormone called glucagon, and you're going to start breaking down those storage molecules. So you're going to glycogenolysis and glucogenolysis, and you're going to release that energy that was stored up in the sugar. And that has to do, the different hormones balance out. So you've got these two negative feedback systems, all right? I also have little notes of the difference between type 1 and type 2 diabetes, right? So type 1 diabetes, the body is not producing insulin or very little insulin, because it's actually an autoimmune reaction where you're killing the cells that produce insulin. Type 2 diabetes is the one that people have a lot. And that's when your body is producing insulin, but you've just got so much sugar in your blood that it just doesn't respond anymore. So that would be the difference. The other thing that I'd like you to think about in terms of regulation is the dichotomy between when you're sick, you have chills and fever, right? And it sounds like a paradox, right? Is it positive or negative feedback? So basically what's happening, right? So your body has its temperature set point. So think of that fish tank thermometer, but your set point is 37 degrees Celsius, right? And what ends up happening is that when you're not well, your body temperature is going to go up, all right? How does that happen? Well, basically, macrophage, when they encounter some sort of an infection, they're going to start releasing signaling molecules. And these things are called cytokines, and they travel through the body, and the brain then responds by raising your set point. So it raises the temperature up, okay? So when that happens, your body is going to feel cold, and you're going to start to shiver, just as if it was cold out, right? So you're going to start shivering. And then when the infection clears, the white blood cells are going to, slow down doing what they're doing. The cytokines are going to go down. Your thermometer in your brain is going to, the set point is going to go back down to 37 degrees. So it may have gone up to 38, 39, depending on the fever. It goes back down, and then you feel cold. And then, of course, you start to get, then you start to feel hot. I'm looking at chills. So then when the body set point goes back down to normal, you feel really hot and you start to sweat, all right? Now, sweat then helps to cool the body off. And we'll talk about in chapter 2 why that works that way, all right? Now, what is the whole motivation behind this? And there's a few ideas. We're not really sure. But you can see them on page 8, top of page 8. So the raised body temperature makes it inhospitable to the infection, right? So bacteria don't like it. That raised temperature may increase the activity of the white blood cells to help you clear an infection. The other thing is that happens when you have a higher temperature is that the liver holds on to iron. And the idea there is that maybe that sequestered iron helps to kill the bacteria because the bacteria would like it, right? So there's these 3 ideas as to how this works, right? Now, there's other things like biological rhythms, all right? Circadian rhythm that affects, your running rate, so it can affect your body temperature, right? So for me, I know I always feel warmer in the morning than I do in the evening, and that will affect also apparently hormone levels. Then you can regulate that, so to a changing environment, right? Spring versus fall. Southerners versus Northerners. I mean, I'm from Canada, and I know in the winter, Canadians like to go down to Florida and like they're out on the beach in bikinis and stuff. And local people are like out with sweatshirts and jackets because they think it's cold. So there's this kind of idea of acclimatization, right? That's also kind of interesting. Now this here gives you your body temperature at different times of the day, right? Your plasma cortisol levels, or cortisol. I have a couple of notes in about cortisol. It's supposed to be the stress hormone, but it's actually also produced to regulate blood sugar, right? So its levels will also regulate sort of cyclically during the day, all right? So it's lowest during sleep and peaks, after waking, right? So it's kind of interesting how that works. So that would be hormone and temperature, all right? Now, the last little part here is on medical imaging. For me, I really just want you to know basically what these do, but it's the important thing to know here is that now, of course, these images are all digital, right? So the computer can store the data, it can be transferred around between different places. When I was young, it wasn't the case. You'd film, and that would record the images, but now it's much easier. So there's like lots of data on this. And why do we take medical imaging? Or why is imaging important? So the idea is to non-invasively image internal structures, and look at the non-invasively also assess function using medical imaging. And there's a lot of different ways to do this. And I have quite a few notes on this. It's not that I want you to memorize all of this, okay? I don't, but I used to work with radiology when I was doing some research, when I was doing clinical research. So I kind of, for me, this is interesting. But really what I'd like you to know are the things that are, in the boxes, like the pros and the cons and what you're using, all right? And very often you use more than one type of imaging to get the information that you want, right? So the first one is X-ray, because that was like really the first one. So what you're dealing with here is X-ray, which is radiation. Basically what happens is the X-rays are shot out at the specimen, and the object or the specimen that's in the way will absorb or allow the X-rays to pass through depending on what they're composed of and how dense they are. So where the X-rays are blocked is going to be lighter part of the image, and where they get through, it'll be darker. So that's why it's really good to look at bone, right? So that's obviously one of the first ways X-rays were used. You can also use sort of like low power X-ray. So look at breast. This is what we do with mammography. And I want to see the very difference between these breasts, right? So here's an extremely dense breast, heterogeneous, average, and fatty, all right? And you can see this, and to be honest, radiologists didn't really understand what's responsible for all of these things. I used to work in a lab, and there was a postdoc there, and that's what his project was to understand what causes the different lightening and darkening in tissue-wise, so that people understand it. But basically what they're looking for is a brightening, which would be a tumor. And the reason you have to go every year after a certain age is because they're looking for changes in them from year to year, okay? But this is like a low energy X-ray, so it's not as damaging, which is the con of X-ray, because it can damage tissue, so it would be cause cancer, which is what you don't want, but you just lower the strength. It's also used for looking at bone density, right? I don't understand it as well, but they look at certain regions and monitor how much of the signal gets through. So the denser, less of it will get through. So this is sort of, there's these thresholds in the particular regions of the body that they use to study that. So that would be bone density scan. So the pros, it's cheap, quick. Cons, damaging, right? So like damaging to DNA, you don't want to cause cancer. Then there's CT. CT commuted tomography. It's also X-ray, but it's high energy. And the other thing is it's not just on one face. It's like a series of X-rays that are done high power, and they take it through the body at different angles. And then with that, they use it to assemble a three-dimensional reconstruction of that structure or the body that you're looking at, right? So they could be done here, like, linearly and take different slices, or it can rotate. So there's different ways of doing it, and you can see the types of images that you get. It gives you a bit more of a readout as to what's happening. But again, you don't want to do it very often because this is not high power X-ray. So that's sort of the con. But the pro, again, it's cheap and quick. But here you got 3D imaging, and here you can also look at soft tissue, not just dense bone. All right. The cons, of course, high dose of X-rays, which would be the same con with regular X-ray. All right. Now, conversely, ultrasound. All right. So here, notice I'm showing the breast again because someone like me, I'm gonna be personal here. My breasts are very dense. So if you look back here, so when I get my mammogram done, I also have to get an ultrasound done. All right. Basically what an ultrasound is, all right, so this is a sonogram, all right, so what an ultrasound is that it uses high frequency sound waves. You can't hear with your ear, right, but they're high frequency sound waves, and the emitter and the detector is the same. So it's basically the sound waves hit things and then they bounce off and they go back to the detector, right? So sort of like an echo, right? So sometimes it kind of echoes, so it goes down, it sort of bounces off, comes back, right? And then you can get the topography of the tissues that you're looking at, or the baby, for example, right? And it's the same thing if you watch those movies where they have those ships and they're like doing it, like, the sonar, and it's hitting off the bottom of the ocean where they're looking for somebody, right? Or they're looking for the Titanic or whatever. It's the same idea, all right? It's a very similar idea, all right? Then there's Doppler ultrasound, where they actually can look at the flow of fluid. So here through the heart, you can see the chambers of the heart and the flow of blood through there. And they do this for my daughter because she has an issue with one of her heart valves, actually pulmonary stenosis. And they look at the flow and how it's working and the ability of the valves to function properly. So here they can see, monitor the movement of the fluid through the structure. All right. So the pro is that it's totally safe. It's cheap. There's no dyes or anything that's involved. The cons are that you kind of have to have some training to understand how it works. I mean, now you can get these beautiful images for babies. Cute, right? But most people, when they go and get an ultrasound done with the baby, it looks like an alien. It's kind of hard to really understand what they're looking at. But if you're a trained tech and radiologist, you can actually look at the vertebrae, make sure they're right, and make sure the different structures are right. You get some training, right? Here, but even I can tell this is a hand, right, and stuff. But there's different. It's not as easy to see the image. Whereas if you're like back here, it's like I could tell these are blood vessels and this is intestine. And the thing I didn't mention here is sometimes they make you ingest some sort of a dye, like barium or something, that will give you even more, we call it contrast, so that you can sort of see things better. They don't do that with ultrasound, though, okay? And then there's this thing called PET, or positron emission tomography. Now, in this case, we're using some radiation. This is not radiation that you're going to explode, right? But basically, what it's really good for is to monitor metabolism, right? And there's different types of, it's a type of nuclear imaging. So it's like a low dose of a molecule that's radioactive. And we'll talk about what that is in the next chapter. But it doesn't mean that it's going to give you, a third head or something like that. There's different types of radiation that can given off by these molecules. But basically, they incorporate these elements into a normal structure, say glucose. So they might use a radioactive form of carbon in the glucose as an example. And wherever you're going to metabolize a lot, you're going to use up a lot of sugar, and there's going to be a lot of that radionucleotide in that area. And this is really good to look at tumors, for example, right? Because tumors metabolize a lot. And then you can get the signal that's given off is from the radiation. It's a type of radiation that's given off, and the detector will detect that, all right? So if it's alpha alpha waves or whatever, right? So sometimes these positive particles are coming off, positrons, all right? So that's what we call positron emission tomography, all right? So that's basically alpha radiation that's coming off, right? And they can also produce other ones that are not as helpful because they might be damaging, but usually they'll use one that's not going to damage. Um, and it's very useful to look at metabolism. So that's the point here. So the pros is that it's safe, and it gives you metabolic information, which you don't have from the other types of imaging. The cons is that it's expensive to make these molecules, all right? And the scans take a while to undergo. So the patient has to be pretty still for like an hour as you're in and out of the scanner to do this. There's a variation of that called SPECT, which I wrote in my notes, is a single positron emission. And in this case, photons are emitted instead, so it would be a different type of radiation that's being given off. Photons, of course, would be light. It's going to be, safer, right? So that's what's going to be given off from these tracers, these radioactive tracers that are used. Okay, so then the next one is endoscopy. Here's where you're really just inserting a camera in some way, all right? And I've got some videos here of vocal cord movements. You can look at that. But it gives you very different information, right? So here you can see like histology versus what you would get of the same place. So if you use these, endoscopy, you can find this, for example, would be colonoscopy. You can see where the polyps are. You can actually sample them and then go and look at what they look like histologically. But here's with the camera there, or this could be down the throat. I don't remember which it is. But you can basically, put this in here. There's other ones, these things like pill cameras, you swallow it. Colonoscopy, of course, goes through the anus, and people don't like that. So there's those different aspects of that. So that's the problem with endoscopy, because it is kind of invasive. It can be kind of invasive. And that's sort of, the reason that, in terms of medical imaging, you do, because you don't want it to be invasive, but it is in this case. But you can see you get these, a lot of information like this. Now there's different types of endoscopy, and I have a list of them on page 10. I'm not going to expect you to memorize these, all right? But you can see the different types of them, of the different types of scopies. So like colonoscopy, arthroscopy, and so on, laryngoscopy, right? And you can see all the names there to see, what there are. So laparoscopies in the abdomen. So the different types of endoscopy indicate, where the camera is being inserted, all right? And because people don't like colonoscopy, there's what we call a virtual colonoscopy now, but really that's x-rays. So this, computed tomography colonoscopy. You still have to prep though. I don't know if you've ever had a colonoscopy, but the prep is a bitch, pretty much. I don't know if they've gotten it better, but I've had 2 or 3 colonoscopies, and that's because, man, but that's the part that really sucks, but I'll just be honest. So the pros are, it can be comfortable. I don't know if I quite agree with that. Well, so the virtual colonoscopy, the pros are that it's more comfortable, no sedation is required, and you don't need to use barium at all either. Not that you're using a colonoscopy, but you don't have to, what you do with other things. The cons is that you can't sample and remove polyps if you're doing a virtual colonoscopy. So you have to follow up with a regular one if you have observed something. Then small polyps might be missed, right? Versus regular regular endoscopy. The pros, obviously, real-time images. You can biopsy directly. The cons, of course, you need good training. Prep can be difficult on the patient, and it's invasive. It's uncomfortable, right? People don't like it. So that's the thing there. Here's a few new things. Here's the virtual colonoscopy. That's why I was like, okay, so you can look at it supine or prone, different ways of looking at it. But again, you can't sample. But yeah, you still have that prep, but it's just much, it's kind of anyways. Um, the other kinds of imaging that's new would be like this thing like motion microscopy, a motion microscope. It sort of can look at, basically looking at the flow of, learning about the flow of blood through the patient's face by looking at the motion of the skin. And to you and I, when we just look at that, we won't be able to tell. But the microscope, this imaging mechanism, so I can't remember if when it's, this is what, you know, static, and then here it starts to look at it. So you can sort of see, you can look at this video, it's kind of cool. But basically you can knows when the heart is beating based upon sort of the color that's given off as it senses the movement, the slight movement of the skin. They're thinking this would be a great way to monitor people really without touch in babies as well, right? So you can monitor their heart rate and stuff like that. So it's kind of interesting. It's not in the clinic yet, but it's something that they're looking at. And I don't know, if you guys are planning on being nurses, you know, maybe that will eventually show up. This one, clarity, this is really cool. This is something more used in the lab. But basically, you can get high detailed molecular and cellular information in 3 dimensions in actual slabs of tissue. You can watch the video and learn about it, but basically they take out the fat and maintain the cells in a gel. And then they can go in with special markers and find out exactly how all these cells interact. And it's really, really super cool. If I would normally have a normal class, I would show the video. So I would suggest watch it. It's super cool. But again, that's not something that you're going to see in the clinic. Now, something you will see in the clinic is MRI, so magnetic resonance imaging. It used to be called nuclear magnetic resonance imaging, and it freaked people out because they thought it was hot. It's not. It's just that the nucleus of the atom, we're talking, or the nucleus of a molecule. I mean, it's kind of hard to explain, but anyways, basically, MRI is really great for soft tissues, right? Really, really great for soft tissues. Fantastic 3D imaging. It explains here in my notes how it works. You don't need to know that, but basically you're putting a magnet and the protons align to the magnetic field, and then it's relaxed. Like when you turn off the magnetic field, and the protons then, what we say is called relaxing, and they go back to their normal. And the time it takes for them to do that is different depending on what's around those protons. And you can detect it because when this happens, there's going to be radio waves that are released, and the detector senses that. And then, like I said, structures around that are going to affect the rate at which it does this. All right, it's really hard to understand unless you're really not in it to understand how it works. But suffice to say, beautiful, beautiful images of soft tissue. And so for me, actually, for my mammogram, I could also go and get an ultrasound done. I said ultrasound, an MRI done. So one year I had the X-ray, and then I had ultrasound, then I had an MRI. Now, the reason it's so noisy, now this is the con, right, is that it is really noisy. And the reason it's noisy is because the magnet is being turned on and off very quickly. So that's what that really loud clicking noise is. The other thing is that it's in a very tight space, and the patient really shouldn't move much. Okay, so that's sort of the problem with that. You can add contrast agents. You don't have to, but you can if you really want to see good images. So there's different types. Since it's a magnet, things that do produce good contrast is iron, and it's really good to look at blood flow because of the iron sensitivity. Another one that's injected is gadolinium, and I've had that done when they were looking at my crazy kidney. The other variation on MRI is what's called functional MRI, and this looks at, and this is when you see when they're talking about brain imaging, when someone's thinking about a certain thing, a certain part of the brain will light up. So this really, what it does is it's really good at looking at flow of blood. So it's going to look at the oxygen levels, and this sort of is going to tell when what part of the brain is most active for different things, right? So you can see the difference between a healthy brain and a patient with schizophrenia, for example. Different parts of the brain are lit up in different ways, all right? So that's sort of cool. You can also use MRI to guide biopsy specimens now too. We usually use that ultrasound if they want to do that. Then, of course, here, like you can see that, so here, MRI localized biopsies reveal subtype-specific differences in molecular and cellular composition at the margins of glioblastoma. I used to study glioblastoma when I was at NYU. So it was really interesting to see this, but you can see that it could be very important in terms of biopsy and localizing where you should take that biopsy. Then non-imaging assessments, all right? Before we had imaging, we had to do things the old school, right? So palpation, so touching body surfaces, listening to the body. Most of the time you just think of a stethoscope, but I was looking at a video the other day and there was a woman, I guess she was I don't think she was a doctor, but she was a nurse of some kind in a country where they wouldn't have all of these fancy imaging tools. And she had a special tool that she put her ear to it and then put it to the abdomen of a pregnant woman. She listened to the baby, right? So listening, percussion, also tapping on the surfaces with the fingertips and listening for echoes, feeling for changes, very, very important, right? So they're old school, but that is other ways of assessing patient health, all right? And that's it for this chapter. So hopefully that's helpful. And again, make sure you go over the PowerPoint and the slides and the video for Atlas A. I called it really anatomical terms and directional terms to start mastering that. And then again, focus on the study guide. And so you know which ones that I really want you to know. It's quite a list, but you're now in A&P, so it's time to step it up and learn all of these things. The sooner you learn it, the better, and you'll forget some of them, and then some of them will come back, right? So, but if you don't start, you're not going to master it, all right? So that's it then.